And it appears that we are officially live. So hello, everybody, and welcome to another session of our Sussex Vision Seminar Series, as always within the Worldwide Neuro Initiative. I'm George Kafetsis, a master's graduate from Thomas Euler's group, and currently a PhD student with Tom Baden. And as your host for today, I would like to once again begin by thanking Tim Vogels and Panos Bozelos for putting forward this ever-expanding initiative towards a greener and much more accessible seminar world. Having said that, allow me, of course, to get back to the reason we all gathered here for today and introduce our guest from Scuola Internazionale Superiore di Studi Avanzati in Trieste, Professor Katja Reinhard. Katja studied biomedical sciences for her bachelor and master degrees at the University of Freiburg and Bern, respectively. She then moved to Tübingen and the Center for Integrative Neuroscience there for her PhD in the lab of Thomas Munch. And during her time there, she studied the properties of retinal codes, both in physiological conditions and disease, and also in a number of different species, providing the first characterization of the output of the human retina on a cellular level. And while maintaining an ethological and comparative perspective from the retina and her PhD to postdoctoral years with Carl Farrow at NERF in Leuven since 2016, studying circuits in higher brain areas, superior colliculus specifically, circuits that receive retinal input and underlie innate behaviors. At the end of last year, Katja moved to Trieste and SISSA as an assistant professor and started her lab there, the Lab of Flexibility in Circuits and Behavior, with the aim being to identify how information about the environment or the organism's state can adapt decision-making at the behavioral level. So in her talk today, entitled Routing and Modulation of Retinal Input to Circuits Underlying Aversive Behavior, we will be hearing about their latest and I'm sure exciting findings. So without any further ado from my side, please all welcome Dr. Reinhard. Katja, the stage is officially all yours.
Thank you very much, George. I will try to share my screen. Can you see this?
Yes.
Perfect. My laser pointer. Okay. Well, thank you very much, George, for the nice introduction, and thank you to the Baden Lab for having me here today. Both during my postdoc and now in my own lab, I'm interested in understanding what happens, for example, in this mouse, in this picture, when it sees danger, like this attacking owl, and reacts to such danger. So my talk today, as George already mentioned, is about routing and modulation of retinal inputs to the neural circuits that underlie these kind of aversive behaviors. I want to start, however, not with a mouse video, but with this video of a little iguana. You see this iguana on the beaches of the Galapagos Islands, I believe, and there's also snakes, and I'll just let you watch for a moment what happens. Right. I really like this video clip, not only because the behaviors are very impressive, but in particular because it illustrates very nicely what we mean when we talk about innate behaviors. This iguana is newborn, it's freshly hatched. It has never seen a snake before in its life. And as you could see, there was no parents or siblings or any other iguanas around that could instruct it on what to do. Nevertheless, it had a very clear program, apparently, in its brain of how to see a snake, recognize that it's dangerous, and react to it. And what's also interesting about it is that it shows different types of innate reactions. In the beginning, it's completely still, observing its surroundings, just sitting there. And then there's a sudden moment where it switches and escapes as quickly as possible, and all the snakes are following it. What my lab here at SISSA is interested in is to understand how such context-dependent changes of innate behaviors, like switching between freezing and escaping, is mediated by the brain. And we have different types of broad questions that we want to address in this context. First of all, what aspects of behavior can be modulated by the situation? Are there aspects of behavior that are invariant? But also, what aspects of visual encoding can be changed by context? Does the snake, in terms of neural activity in the retina or further downstream, look the same in these different situations, or can this visual representation in the brain change depending on the context of the animal? We also want to know whether changes in behavior, so switching between freezing and escape or other changes, are encoded already in the very early stages of these visual motor circuits. And finally, we want to find general principles across contexts and species of how flexibility can be achieved by the brain. Today I will speak about two projects from my postdoc in the lab of Carl Farrell at NERV, where we built the basis basically of what my lab wants to do. And we asked the question of how circuits are organized to link information to appropriate behavioral outputs. In a first part, we addressed this by studying how visual information is routed in hardwired circuits. And in the second part, I will talk about how we investigated how brain-wide inputs are organized to modulate this visual motor transformation. In the first project, we addressed this hypothesis that circuits mediate fast innate reactions are organized in what we call hardwired architectures. What I mean with this is illustrated in this very simple schematic. It shows how the relevant neural circuits for innate behaviors are organized in parallel labeled lines that connect very specific information, in this case from the eye, through clearly defined networks of neurons to guide a particular behavior. The alternative to this hypothesis would be that sensor information is broadly broadcasted from the retina, and that you need local computations to extract relevant information to decide if something is a danger stimulus or not, and to then activate the correct muscles. Because innate behaviors have to be so fast and reliable, we believe that a hardwired architecture would be much better at describing them. To test this hypothesis and to measure innate behaviors in general, we have used a very standardized set of visual stimuli consisting of stimuli that mimic an overhead cruising predator, so a bird that flies far above on the sky and which is looking for prey, as well as stimuli that mimic attacking predators, like the famous looming stimulus, where it moves towards the animal of interest. This is an example video of a mouse reacting to such an overhead cruising predator stimulus. I will play it a few more times because it's very difficult to see this very small dot that moves from the left to the right. But this mimics such an overhead predator, and as you can see, when the mouse detects it, it waits for the stimulus to pass by. So we are inducing here an arrest or stopping behavior, which is a quite common reaction observed also by other scientists to these kind of stimuli. On the other hand, in this case, we use a looming or expanding disc stimulus, which we'll start in a second. And this mimics an attacking predator stimulus. And you can see that the mouse here decides to run away as quickly as possible to the shelter at the other end of the setup. This is a commonly observed response to these attacking predators, but both in my postdoc lab as well as other scientists have found as well freezing responses to these looming stimuli, depending on the particular setup, context, and so on. For the audience today, I don't need to highlight that any brain circuit that links seeing a stimulus to doing a behavior has to start in the retina. And we know that the retina of a mouse contains over 30, probably rather 50 different retinal ganglion cell types that have been characterized anatomically, functionally, and morphologically. And each of these cell types transmits a different type of information to the rest of the brain, such as movement direction, contrast, size, and so on. One of the main targets of the retina is the superior colliculus, which is part of the midbrain. It has a layered structure and is retinotopically organized. And it consists of different genetically identifiable cell types, of which I'm showing you the four most easily targetable and best described here. We have on the top the NTSR1 cell type, which are the famous white field cell types. This is huge dendritic trees that span up to 60 degrees of the visual field. Then we have parvalbumin neurons, which, similar to the retina, are mostly excitatory in the colliculus as well. We have inhibitory projection neurons, which are labeled in GAT2 animals. And these are anatomically characterized as horizontal cells. And finally, we have the GRP neurons, or narrow field cells, which are another class of excitatory projection neurons. Each of these cell types has a different set of downstream targets after the colliculus, and especially the NTSR one is quite an interesting one because it has this very dominant target, which is the pulvinar, part of the visual thalamus, and optogenetic activation of this circuit tends to induce a freezing response in mice. Escape behavior, on the other hand, is mediated by other circuits, such as the ones that pass through the parabrachydonal nucleus, or PBG, or the dorsal periaqueductal gray down here. This organization of these output circuits of the colliculus reinforced our hypotheses of a hardwired architecture. And to explicitly test this, Jen and I looked at the beginning of these circuits, the visual inputs from the retina. The question we asked was, how do these different output circuits of the colliculus sample visual information from the retina? And we focused on two targets, the pulvinar and the PBG. And we used a dual viral strategy to specifically label the ganglion cells in the retina as part of these circuits. This consisted of first injecting a retrograde virus, in this case an HSV, into one of the targets, for example, the pulvinar. And this virus infects the collicular neurons of projected pulvinar and leads to the transcription of a TVA, a receptor in those neurons. We then injected a rabies virus into the colliculus, which can only infect those TVA-expressing neurons and which can jump one synapse upstream and therefore infect the retinal ganglion cells that form part of these particular circuits. This is an example retinal ganglion cell after this viral strategy. And in total, we labeled almost 600 such retinal ganglion cells that are part of the collicular pulvinar or the collicular PBG circuit. Our goal was then to classify these retinal ganglion cells into the known cell types of the mouse retina. And to do this, we used anatomical parameters such as the area of the dendritic tree, the soma size, and importantly, the stratification depth of the dendrites between the two chat bands. Especially this last parameter was a very important parameter, and we compared it to this beautiful database of the stratification patterns of retinal ganglion cells from this paper from 2018. We complemented this with a few other cell types from other papers on the retina. And the main thing we did to classify our ganglion cells was to compare the stratification depths with the best fit from this database of, I think, 56 ganglion cell types that we had in the end. This classification was also aided by molecular parameters. We stained, for example, for SMI32, so we stained alpha cells, and we use this information to further refine our classification. Having done this classification, we found that out of the over 30, maybe 50 ganglion cell types that exist in the mouse retina, only 14, which are depicted here with example cells, projected these two circuits. And what is more is that out of these 14, only 6, shown here in the center, are shared between the two circuits, whereas the 4 here on the top are specific for the collicular pulvinar, and the 4 here at the bottom for the collicular PBG circuit. This means that we have an only partially overlapping and quite small subset of retinal information is routed in these two behaviorally relevant circuits. And of course, because we did our classification based on these published data, we can put a name to each of these cell types. And I just want to highlight two examples here. In the shared cell types, we have the transient of alpha cell, for example, which is the looming detector of the retina. And here in the PBG-specific cells, we have the on-off direction selective cell, which we do not find at the beginning of the collicular pulvinar circuit. So given this classification and also electrophysiological data from our own lab, we can summarize that the cells that project to the pulvinar circuit respond well to small and slow stimuli, which is good to encode a cruising predator stimulus. The shared cell types contain multiple cell types that respond well to these looming stimuli, so they encode very well an attacking predator. And finally, the cell types down here, which are specific for the PBG, contain the direction selective cells, but also respond to big and fast stimuli. And this might be good to detect a ground predator, where movement direction might be more important than for an aerial predator. So we not only have a small, but also behaviorally relevant circuit-specific set of retinal inputs. We then wondered if the separation of visual information is visible in the downstream targets of the colliculus as well. And so we recorded directly from the pulvinar and the PPG in behaving animals in vivo using neuropixel recordings and while showing visual stimuli to our mice. What I found is indeed that neurons in the PVG and pulvinar have different visual preferences. For example, neurons in the PVG respond very well to big, fast, and small, fast stimuli. But if you look at expansion or looming stimuli, we find very similar responses, and this is also visible in the average population response of neurons in these two nuclei. If you use a moving bar to test for direction selectivity, you find these very interesting cells in the PPG that are direction selective, but often for two directions, and they tend to be a forward movement and a sort of down-backward movement direction, whereas in the pulvinar, we do not find very selective cells. So the example on the right is a representative example of the pulvinar. And of course, you quantify that with a direction selectivity index. And on one hand, you can see here that pulvinar neurons do not respond very well to moving bars, and the ones that do respond are not direction selective, or most of them are not direction selective, whereas in the PPG, the vast majority of neurons is clearly direction selective. So the visual preferences of the pulvinar and the PPG match to a large degree their specific disynaptic inputs from the retina, which means that circuit-specific retinal inputs are passed on to the collicular targets. To give an example, direction selective cells only innervate the collicular PPG circuit, and we find very strong direction selective responses in the PPG, but not in the pulvinar. This also fits the knowledge we have about behavior, where cruising predator stimuli, the small, slow stimuli, tend to induce a freezing response, while looming stimuli can induce either escape or freezing. So taken together, we found that circuits through the superior colliculus have a hardwired character, where visual information about a very specific threat-like stimulus is routed in separate circuits to produce stimulus-specific innate reactions. So far, we focused on the very first synapses of the circuits that mediate innate behaviors, and in the next few slides, I would like to tell you about work where we investigated the brain-wide downstream circuits of the colliculus. And this is a project that was spearheaded by Arno and Anja. Here in the first step, we express channelrhodopsin in the colliculus of different Cre mouse lines that express Cre in these different cell types in the colliculus. And we use the optic fiber to optogenetically activate those different cell types and looked at the behavior that we could induce. It is not very surprising that when we activated NTSR1 neurons, we saw this stopping behavior. So the average speed it goes down to 0 during the activation, and you can see that as well in the 2D locomotion plot over here. And this fits what we know from other studies that have optogenetically activated those pathways and found freezing behaviors. When we activate PV neurons, we find a mix of behaviors where some animals tend to increase their speed and others tend to slow down. And if we activate inhibitory projection neurons, the GAT2 neurons, we see this clear increase in speed compared to control animals, but the animals tend to move in one particular direction. Taken together, this shows that different cell types in the colliculus are involved in distinct behaviors, and the evoked reactions look quite similar to behaviors you would see during visual stimulation as well. So given the results from our retinal input study, we wondered to which degree the brain-wide neural networks that are activated in each of these different situations are similar or distinct from each other. To address this, we used a technique called functional ultrasound imaging, or fUSI. fUSI uses the Doppler effect to measure blood flow in the brain. And to do this, one implants an ultrasound transducer, shown over here, over the brain. And we can cover about one hemisphere in a mouse, and we can still combine this technique with optogenetic activation of the neurons in the superficial superior colliculus. This leads then to anatomical maps, like you can see over here, which indicate changes in blood flow compared to a baseline. This is an example video of the changes of blood flow when we activate NTSR1 neurons in the superior colliculus, where brown indicates increase in blood flow and green decrease in blood flow. And I don't want you to focus on any specific details. I just want to illustrate with this that there is quite some modulation going on, but there's also quite some parts of the brain that are not modulated in this experiment. And what we can do now is to cut slices through this brain and compare the activation patterns across the different cell types that we activate in the colliculus. And when we do this, I hope it's quite striking for you as well. We see that the activation patterns are very different depending on which cell type we activate in the colliculus, and therefore which potential behavior we are inducing in that particular case. The most striking is the GAT2 condition, where we have very few modulated areas, even though those animals show very clear behavioral output during those experiments. But also between the NTSR and the PV, we can see different areas are activated to a different degree or with different signs. If we group these areas by different regions, such as cortex and so on, we see that uniquely activated areas are mostly found in the prefrontal cortex, the amygdala, and the thalamus, whereas common areas are more common in the hindbrain, midbrain, and hypothalamus. One example of an hub, which is activated very similarly across different experiments, is the zona incerta. The zona incerta has recurrent connections with the colliculus and has been associated with various motor behaviors, for example, movements in Parkinson's diseases, attention, exploration, hunting behavior, and so on. So it makes sense that a nucleus like the zona incerta would be activated in the context of very different behavioral patterns. Another extreme is the anterior cingulate area, or ACA, which is part of the prefrontal cortex, which is activated, the blood flow increases when we activate NTSR neurons. Blood flow decreases in this area during parvalbumin activation, and there is no modulation in the GAT2 experiments. It's not entirely clear what the role of the ACA is in the context of innate behaviors, but clearly it is very differently modulated depending on what circuit we activate and what behavior is induced. This suggests that not only the very first synapses, but the whole neural network that is activated during a particular stimulus or behavior is relatively sparse and specific to that combination of stimulus and behavioral output. So I hope I could convince you in this first part that there's a clear structure to the circuits that connect visual information to motor outputs. And this work so far focused on the behavior driving circuits, so the circuits that detect visual information and activate brain areas that eventually induce the behavior. But in the second project, we asked how non-retinal inputs can modulate and adjust this visual motor transformation that is mediated by the circuits of the first project. And to address this, Chen and Norma focused on the GAT2 cells, which are the inhibitory projection cells of the colliculus and which have two main targets, the parabigemal nucleus, or PBG, and the lateral geniculate nucleus, the LGN. The first question we asked here is whether GAT2 neurons form collaterals to project to these two different output areas, or whether there's two different populations of GAT2 neurons depending on their projection target. And to answer this question, we used again retrograde viruses that express different fluorophores and were injected into the PBG and LGN in a GAT2 Cree mouse to specifically label the GAT2 neurons that project to each target. When we look at the superior colliculus after these experiments, we see that cells usually either express mCherry or GCAMP. And indeed, if we statistically analyze this, we find very few cells that have both projection targets. So GAT2 neurons do not form collaterals, but they're really two different populations that project either to the LGN or to the PBG. Given the results from our first project, of course, we wondered whether these two different populations of GAT2 neurons would receive different visual inputs from the retina. So we repeated our experiments with the retrograde injection and the rabies injection to label specifically the retinal ganglion cells at the beginning of each of these two circuits. However, differently to what we found for the pulvinar and the PBG, we find here that the retinal ganglion cells are quite similar at the beginning of these two circuits. They first of all have very similar sizes of dendritic diameters. They also have similar proportions of different cell types. So we have similar proportions of on-off direction selective cells, alpha cells, and FOXP2 cells. And if we record visual responses in these neurons, we find similar sets of visual responses. So we find the clear on-off DS cells, responses in both of the circuits. We find non-direction selective cells with different transiency in the two circuits. We find very similar looking responses to different looming and expansion disc stimuli. And we find typical responses like transient ons and transient off responses at the beginning of both circuits. So given that they have similar retinal inputs, we presume that GAT2 neurons have similar visual preferences, but we wanted to specifically test this. So we injected viruses to express GCAM6 again in circuit-specific GAT2 neurons. And we placed our animals on a floating ball, and we used 2-photon calcium imaging to record the visual responses of these GAT2 neurons while showing visual stimuli on a screen to the mice. And as you can see here, the overall pattern of visual preferences in these two circuits is very similar. So we showed looming stimuli of negative and positive contrast. We had receding and dimming stimuli and small discs while moving slowly and quickly with different contrasts. And for each of these stimuli, the overall preference of GAT2 neurons is similar, independent of whether they projected the LGN or the PBG. So taken together with the retinal data, we can conclude that GAT2 visual features are circuit independent. Okay, so GAT2 neurons received similar visual inputs, but nevertheless, they projected different targets. And they wondered what their role would be in innate behaviors and whether they would have a different role in innate behaviors. And we decided to test this chemogenetically. So we inhibited the projection-specific GAT2 neurons by expressing DRETs in these projection-specific neurons. And afterwards, we injected the ligand for those threads, CNO, and we exposed the mice to a looming stimulus and observed their reactions. Just to briefly remind you, control animals tend to respond with an escape response. So this is a control mouse after looming stimulus. And you can see that as well here, the average speed trace is a slight increase during the looming stimulus. And overall, we see that about 60% of our animals are escaping and the remaining ones are freezing. Now, if we chemogenetically inhibit GAT2 neurons that project to the PBG, which means that we remove inhibition from the PBG because we're inhibiting inhibitory neurons, we see a change in the probability of escape responses. Now we have about 80% of our mice escaping, almost none of them is freezing, and we also see that this escape response seems to be a bit faster on average than in the control animals. So we see an increase in escape behavior if we remove inhibition from the PBG. On the other hand, if we remove inhibition from the LGN, we see quite the opposite, where now freezing is the dominant behavior in those animals. And we see that as well in the average speed trace, which is dominated by those stopping or freezing behaviors. So these thread experiments suggest that GAT2 neurons have circuit-specific effects on behavior. And you can see, sorry, I forgot to show you this video. You can see that these freezing behaviors are not only more common, but they're also really strong. So we're really increasing very much the freezing patterns when we inhibit inhibitory neurons that project to the LGN, and we see the opposite for the PBG. But because we showed that these GAT2 neurons receive similar visual inputs, these specific effects cannot originate from their retinal inputs, but are likely due to non-retinal modulatory inputs. And so, of course, when we did our rabies experiments, we not only labeled the circuit-specific retinal ganglion cells, but all the brain-wide circuit-specific inputs, because the rabies just jumps one synapse upstream and infects any neuron that projects to your TVA-expressing cell. This allowed us to look at the input patterns across the whole brain and to count rabies-expressing cells in each of the brain areas. So here you see an example of the zona incerta with all the rabies-labeled cells of the zona incerta. And if we do this for all the brains for the two circuits from multiple animals, we can see just by looking at the overall regions, cortex, subcortex, and so on, that there's difference in the innervation patterns for the two different GAT2 populations. For example, cortical neurons tend to innervate ipsilateral GAT2 neurons that project to the PBG, whereas for LGN projecting GAT2 cells, the cortical inputs seem to be more similar distributed across the two hemispheres. We express these preferences for different inputs with a bias index, which you can see here for all the areas that provide inputs to GAT2 neurons. And you can see on the left side in blue, the areas that provide stronger inputs to LGN projecting GAT2 neurons, and on the right, the ones that have a bias towards PBG projecting GAT2 neurons. And one example, for example, on the blue side, so LGN preferring is the anterior cingulate area again, which I mentioned before, where you see very strong labeling if we backtrace from the LGN projecting GAT2 neurons, but very few cells in the other case. A more moderately biased area is the zona incerta, where you have quite strong labeling in both cases. And on the other end of the extreme, we have, for example, the somatosensory cortex, which provides almost exclusively inputs to the PBG projecting GAT2 neurons. We can also group those areas by their main function, and we find that PBG projecting GAT2 neurons receive more motor and touch-related inputs, whereas there's a small bias towards more vision-related inputs for the LGN projecting GAT2 neurons. And we can actually see this bias in the population response of GAT2 neurons in our GCamp experiments, where we did calcium imaging in freely behaving animals. And we can compare how strongly GAT2 neurons are modulated by visual stimuli, here on the y-axis, or locomotion on the x-axis. And you see what one would predict from the brain-wide inputs with a stronger modulation by locomotion for the PBG projecting cells and for visual inputs for the LGN projecting cells. So clearly this is a substrate to enable circuit-specific modulation of GAT2 neurons and thereby of the induced innate behaviors. And so to specifically test whether such non-retinal inputs do impact innate behaviors, we test the role of two such inputs, the ACA, here we took together the ventral and the dorsal ACA, and the zona incerta. We again used our rabies virus approach to express channel rhodopsin in these circuit-specific inputs, including the ACA and the zona incerta. And we then exposed our animals again to the looming stimulus while also optogenetically modulating one of these input areas in a circuit-specific way. Before I go to the results, I want to briefly remind you of our DREAD experiment results. When we removed inhibition from the PBG, we saw an increase in escape, while removing inhibition from the LGN increased freezing responses. And if you looked at our control animals, they generally respond very well to looming stimuli, and the majority, around 60%, responded with an escape behavior. Now what happens if we optogenetically modulate those inputs? If we do that for the ACA, in the case of the PBG circuit, what we're doing really is activating excitatory pyramidal cells. So we're increasing activation of GAT2 neurons, which means we increase inhibition of the PBG. And as one would expect from this, we see an increase of freezing behavior or a decrease in escape behavior in those animals. On the other hand, if we modulate GABAergic inputs from the zona incerta, we decrease the activity of the GAT2 neurons. So we do something similar like in the DREAD experiments and remove inhibition from the PBG. And indeed, we see now a decrease of the freezing responses and increase of the escape behavior in those animals. If we do the same thing for the LGN projecting GAT2 neurons and we modulate the ACA inputs, we see, to our surprise, that the animals actually stop responding to looming stimuli. So we cannot classify them into freezing or escape because they simply don't react to looming. And we're not entirely sure yet why this is the case. One potential reason is that these inputs are extremely strong from the ACA. So it might just be creating so much inhibition in the colliculus that nothing can happen anymore. Or there might be collaterals from those ACA inputs to other areas, to other cell types in the colliculus or somewhere else that could counteract any influence that it has. So this is an open question still. On the other hand, if we do the same experiments for the zona incerta inputs, which means that we are removing inhibition from the LGN, we still have less animals that respond, but the ones respond do it in the way that we expect, with an increase in freezing behavior, as predicted by the DREAD experiments, for example. So taken together, we found that activation of circuit-specific non-retinal inputs can change the balance between different possible innate reactions in a rather predictable fashion. So we can increase or decrease the fraction of animals that escape or freeze, depending on whether we increase or decrease inhibition in the PBG and the LGN. And to summarize and come back to our initial question about how circuits are organized to link information to appropriate behavioral outputs, what we found here is that circuits that mediate innate reactions are organized to link very specific stimulus information with the appropriate behavioral reactions. However, some stimuli can activate multiple behavioral pathways, for example, the looming stimulus, and here contextual information might change the balance between the different possible outcomes. And indeed, we find that non-retinal inputs are also organized in a circuit-specific way, and hence have the potential to route different aspects of contextual information to different behavior-mediating circuits, and therefore change whether we see an escape or a freezing or something else. And just to give you a very, very brief outlook, my lab now is building on the circuit structure to investigate how selected contexts act on innate behaviors. There's different classes of contexts that require adaptation, which can be transient or permanent, as well as internal or external. And this means that contexts range from habitats over food to light availability, stress, congenital disease, and all of them can change how we react to the same visual input. And we are now addressing in my lab specific contexts from each of the quadrants here to understand how these affect visual motor processing and innate behaviors, and eventually hope to understand where and how adaptation is introduced in such a reliable stereotypic system. And this also goes beyond how we can acutely integrate contextual information, but also addresses the question whether an escape response, for example, produced in a different situation or produced by a different species, is mediated by the same neural circuit. But this is hopefully the content of a future talk. And for today, I would like to end by saying thank you to the Faro Lab, especially Carl, Norma, Arno, Anya, and Jen, our collaborators at NERV for the fUSI experiments, Anna and Gabrielle, our former students that were a part of these studies. And if you would like to know more about my lab or work with me, you can come to my homepage or send me an email. And thank you very much for the invitation today.
Thank you very much, Katja, for this very interesting data and the fantastic presentation of how you try to go circuit by circuit and, you know, isolate the effects in the behavioral output that you observe. If you would like, you could stop screen sharing so we appear
Sure.
concurrently at people's screens. And as we are waiting for some questions, I will very shamelessly start asking my own while
Sure,
I'm also posting the Zoom room link there so people can also join us for the offline get-together that we will have. So the first question that I have is like not related to the retina part, but to the second part of your talk that you mentioned that you have these two different populations of GAD neurons. And I was wondering whether the neurons that project to the pulvinar, if you would expect to have connections within the superior colliculus, like different connections between the two subpopulations of GAD neurons. Like, is it known, or could you speculate something on that? If the neurons that project to the pulvinar also project to the GAD neurons?
You mean the NTSR neurons, if they
yes,
also
the first ones, yeah.
If they have interconnections within the colliculus, basically, is your question.
Exactly, because are these also inhibitory neurons, or are they excitatory?
The NTSR are excitatory neurons.
Excitatory. So I was wondering whether with different projections to the different subpopulations, they could regulate whether the animal will freeze or run away.
There is definitely some interconnection within the colliculus, mostly probably through inhibitory interneurons, but there is still, you know, a lot to be done in terms of how they're actually connected with each other. We know that, for example, there is inhibition of large stimuli on the level of NTSR neurons. So if you look at the retinal inputs, they respond, for example, to full field chirp stimuli, but the NTSR neurons or the downstream neurons in the pulvinar, they don't respond to full field stimuli. So somewhere there is local inhibition, you know, that removes certain information and that shapes again visual processing. Right. To which degree that can change the behavioral output, we don't know yet.
Yeah, I was just wondering about latencies and, like, for example, when you do the behavioral recordings, are you quantifying, or is it possible to quantify the response latency to a looming stimulus, for example? And like,
yes, we do that.
Whether with your manipulations this changes differently, like depending on whether the animal is freezing or escaping?
We haven't looked into much modulation within a behavior yet. We're looking at that in terms of changes with ambient light, actually. So if you change the ambient light, the latency of escape responses changes as well. But we haven't looked at it in this particular context too much. They're definitely more vigorous, right? You saw the escape response, if the PBG is less inhibited, is stronger, and I think it starts earlier, if I remember correctly. But we haven't looked too much into those aspects.
Right. Thank you very much. And going towards the retina part of your talk. So when you have the viral injection and you trace the signal back to the retina, do you observe, like spatially speaking, a different profile? Like, do you label more neurons, I don't know, at the part of the retina that looks at the sky, or?
Yeah, we actually tested for that, and we found a very homogeneous distribution of the inputs. So neurons in the colliculus that project to the pulvinar receive inputs from the whole visual field, as far as we can say, which makes sense because these circuits are not only involved in escape and freezing, but they're also involved, for example, in hunting behavior, where the stimulus would come from below. So the stimulus might be processed differently where it is in the hemisphere, but all the circuits that we looked at receive inputs from the whole visual field. And
like, I know that you didn't check for physiological data from the retina, but when it comes to the on-off DS, you could expect, again, like a fair, unique, like a uniform representation of the different direction preferences.
Yes, I don't think we tested whether that was uniformly distributed in that project. So in the PBG, for sure, it's not, as I showed. So these cells have certain preferences, but we didn't check if that's true in the retina as well.
Yeah, and the last thing I have as a question, like, and if you could comment on it. So I find it quite interesting that the neurons that project from the retina to the pulvinar, you have the F mini off neurons, if I remember correctly. And I think the Jacobi and Schwartz paper has shown that they are like very specifically tuned to to specific sizes and motions and so on. And they show some direction selectivity as well, right? So even though you don't have the traditional on-off DS projecting to the pulvinar, like to the neurons that project to the pulvinar, I'm sorry, you have this F mini, but then you didn't observe any DS profiles, right? That's true, but as far as I understand the F mini data, this is very specific to a certain light level where they turn direction selective due to the inputs from different bipolar cells, but it's not a general feature of them. Right. And like, I guess your stimulus arsenal could be excluding some very specific activity of these cells. Yes. I mean, we did physiological recordings in the retinal ganglion cells, not in all of them, but in a subset of them. And we didn't see DS responses at the beginning of the pulvinar circuit. I see. Okay. Thank you very much for that. Let me check again in the channel. Yeah, so given that there are no questions appearing and maybe people are kind of shy and they would like to join us offline to ask, or then maybe they don't want to reveal, grand application ideas, I will probably terminate the broadcast and see any interesting parties from the audience at the Zoom room that you can tune in by clicking on the link. So thank you very much once again, Katja, for today's fantastic talk. Thank you.